Well, thank you for having me here, and nice to meet you all. My name is Adrià and José, and I'm going to be presenting some work we've been doing regarding Polycystic Echinococcosis in Panamazonia. And this is already published in a paper in PNAS. Here you have the link in case you want to take a look into that. It's going to be obviously better explained than what I'm going to be able to do here in 15 minutes and so on. But let's start. Polycystic echinococcosis is it's a neglected zoonotic disease. It's a parasitic disease. It's called by a cestoid called Echinococcus bogeli. This disease is potentially deadly among humans. It has a case fatality rate quite high of around 30%. And it naturally circulates in Panamazonia thanks to a two-host sylvatic cycle where wild dogs can act as final host, and some rodents, especially the Cunuculus paca, can act as intermediate host, and thus allowing the parasite to complete its life cycle. The thing here is that the hunting of the Cunuculus paca, and the paca is a very appreciated and heavily hunted animal, allows this disease to sort of establish a domestic transmission cycle where domestic dogs can act as final hosts and substitute wild dogs, and humans can act as intermediate hosts, then allowing again the full cycle of the parasite. As I was telling you, this is a neglected tropical diseases, but there are different sort of also neglected tropical diseases, and this is one of the most truly neglected ones. There are only around 200 human reported cases in history, which is the evidence that we have here. Obviously, it circulates silently, but these are the reported cases. Here you can find the evidence that we have in these purple points of human infections, and we also have evidence of infections in animals, especially in pacas, that you can find here in blue. Obviously, well, needless to say, this is Panamazonia with all the different countries of the region. And this data is overlaid on the presence of these intermediate and final hosts. This is very rich data that will help us understand this disease better. So, what are the objectives of our research here? Basically, to answer two open questions. The first one is, which is the spatial distribution of the disease? This is still an open question. As I was telling you, this is a truly neglected disease. And second, in a more maybe scientific fashion, to try to understand which factors determine environmental factors, climatic factors, cultural factors. No, what of these factors can help shape the spatial distribution of the disease? And here we'll pay attention because we will separate between the sylvatic circulation of the disease, so how it naturally circulates in Panamazonia, to the spillover of the disease, so where it jumps to humans. And finally, obviously, we will be able to answer the final question, which is whether this is a climate-modulated, climate-driven disease, and if this is the case, which is the mechanism by which this takes place. So what we did is to run two separate models, as I was telling you before, and given that our data, or let's say we have data of human infections and of animal infections separately, we decided to run two different models, an animal model, animal infection model, and a human infection model, to see how different covariates affect the process, like different stages of the transmission process. Temporal information here was very scarce, so we couldn't approach this with typical time series or approaches or other time-aware statistical methods. So we had to move to spatial models, and especially we chose maximum entropy models for a series of reasons, and two of them are that they only require presence data, so where this disease is present and not absent, which is also hard to establish. And secondly, because they provide metrics or indicators of the variable contribution and response functions. Basically, this means they allow a sort of readability or understanding of the process, which was key for us at this stage. Which are the factors, which are the covariates that we think can influence the disease? Obviously, first of all, the presence of the host, the presence of intermediate hosts, and the presence of final hosts. Third, obviously, the distribution of the human population. We're talking about also human infections. Four, and this is a bit harder to assess, to try to know which is the hunting interest and the hunting pressure for pacas, as this is a key aspect for the disease to jump to humans. And finally, we wanted to explore whether land use change, and climate change could impact how this disease circulates. However, this was more of a wish list of things that we would like to have, not things that are there available for us to use, because only number 3 and number 5 are there available for anyone to use. So before we went to the modeling part of the Maxent models parts, we had to do a lot of previous work. So first, what we did is we applied ecological niche models to try to derive the first 2 covariates, regarding the presence of hosts. For number 4, for the hunting interest in pacas, we used a massive hunting data set that our local co-authors had assembled throughout the past years, and used kriging, so sort of a statistical interpolation, to derive the sort of continuum estimate of the hunting pressure for pacas. And finally, regarding climate change impacts, we use an advanced time series decomposition tool called singular spectrum analysis to derive ourselves some indicators of climate change. I'm not gonna dwell on this because we don't have time, but this is just the same, like how we derived these covariates. This is more like of a graphical summary. Methodologically interesting, so we can talk about this later at some other point if someone wants, but now I'm gonna skip it and go to the next part. Just let me say, and let me focus on this 6 covariate, the climate change impacts. So what we did, we took the time series of the last 30 years of, for example, temperature, and we made a decomposition of this time series on trends, so how it has been changing. And then also in interannual variability, which in Panamazonia is seen to be ENSO-driven. ENSO meaning El Niño Southern Oscillation determines this interannual variation of temperature in Panamazonia, and this ENSO has 2 main modes of variability, which are the quasi-biennial and quasi-quadriennial modes of variability that have different spatial patterns, so we decompose them as well. So for each variable, like for temperature, we derive 3 indicators, trend, QQ, and QB, and also for humidity or precipitation and so on. So finally, everything was ready to run the Maxent models. We had all our covariates, the one we retrieved, the one we built, the training data, and so on. So we ran the 2 separate models. We did this in a rigorous way. We looked over all the key parameters in Maxent, regularization terms, features, and so on. We did an assessment of the skill of the model with a cross-validation. And given that the skill was similar between some of the models, we analyzed the 5 top-ranked models basically all together to make sure we had sound, stable results. So here we have the results of the first model, the animal model. And if we're short of time, let me just focus on panel B. We have the permutation importance. This means how important is each variable in accounting for the spatial distribution of the disease. And let me just focus on the most important variable, which is this QBT, this quasi-biennial mode of temperature, which if we go to panel C, you will see it has a response function which shows a decreasing trend, meaning that a higher QB translates into a lower risk of having sylvatic circulation of the disease, and on the contrary, a low QB means a higher probability of having the sylvatic circulation of the disease. Then we had other functions, land use, natural suitability, which basically account for the common presence of intermediate and final hosts and so on, but I don't have time to comment on everything here. For the spillover model, results were different. We were expecting this, obviously. The most important variable was the distribution of population, which makes complete sense. Humans are the ones that are getting affected. But secondly, the first environmental, let's say, variable was the quasi-quadriennial mode of temperature, like the other one, not the QB like before, now the QQ. And if we look at the response function in panel C, this is the second one here, you see that what we find is that the higher the QQ, the higher it is the probability to have spillover, basically, of the disease. Apart from population and QQ, then we follow with land use, the QBT, and these are the exact functions that we saw before with same response functions, and so on. Well, so given that these 2 variables, this QB and QQ, stood out as very relevant variables, we took some time to try to better understand them. So here you have their spatial patterns. The QB here on the left presents a sort of dipole. We see, like a hotspot around the Guyana shield, and the other dipole maybe being the Western Brazil or also Peru, Bolivia, and so on. Whereas the QQ has a very different shape, and as you can see here. If we take a look at some of the decompositions of temperature, and panel A here, we are focusing in a point in the Guyana Shield, we see that indeed temperature shows this very marked, strong biennial cycle. So every 2 years, temperature goes up and so on. And this is why here we see a hotspot of this QB, and here the QQ, which is the in red here, is not so important in explaining temperature. Interestingly, if you look at the graph below, you will see that these like pulses, these biennial pulses of temperature generate rainfall afterwards in a convective manner, okay? If we now move to panel B, which is like in the other dipole in the southwestern Brazil, what you see is the temperature there is much more stable. There is not this QB cycle, and QQ basically dominates. But the reading also with precipitation is that everything is much more stable, especially if you look at the axis, you will see that the ranges are lower. So then we went on to try to understand how the QQ that only appears in the spillover model, so not in the natural circulation of the disease, can affect the spillover process, and why and where exactly. So we went back to the amazing hunting database that I told you before that our co-authors had assembled during the last 30 years, and we constructed an aggregated time series of hunting practices in Panamazonia that you can see here, and we derived a sort of low frequency time series. And we did the same with the ENSO, the El Nino, as I was telling you. This is what is forcing the climate in Panamazonia, and this is basically the same as the QQ, okay? So we did a sort of analysis on how one of if the ENSO affects the other variable, the hunting variable, and we found very strong associations, statistical associations of how El Nino was affecting hunting practices in Panamazonia. And they were anti-correlated. Throughout the whole period, there was just one anomaly here in 2010. You can see here it in red, but this can be explained because this was a weird year in terms of the Nino, a Madoki type, well, it doesn't matter. But we can also explain this and therefore see that there is a strong anti-correlation between the two variables. So now we go to the last part, which are the mechanisms, the most interesting one. What have we found? Our results regarding the QB, they show that we find the same response function showing up in both models. And we read that as a transfer of information from the sylvatic model to the spillover model. This is natural because for humans to be infected, we need the disease to be there. We need sylvatic circulation in the same places. The way we read that is that Ebogeli thrives in the areas with less QB amplitude. This means in places which are more stable, at least stable in terms of temperature and precipitation. And we just read this as a sign of how this parasite thrives in stable environments. Why is this important? Probably these variables can have a direct effect on the parasite when it's waiting to be ingested and so on the floor. Proper levels of humidity are essential, and excessive humidity or droughts can kill this parasite. And we can have also effects on the host, on their behavior, populations, and so on. Regarding the QQ, this is a variable which was the most important environmental variable in the spillover model, and it was only showing there and not in the other model. What we found is that the spillover risk of Ebogeli increases along increasing QQ amplitude. Our co-authors were always telling us that the Amazon experienced seasonal changes in food sources. Fishing being dominant during dry season because water is low and their fish accumulate and it's easy to fish, and hunting then becoming more prevalent during humid season, because there's too much water, it's hard to fish, and people switches to hunting. What we found, we think, are the same differences, but in an interannual scale. When we have a warm ENSO event, an El Nino event, this leads to drier conditions in Panamazonia, especially in the south, and this favors fishing as the main food source. This results in a decrease in hunting, and if we have a decrease in hunting, we have a decrease in the spillover risk of Ebogeli to human. And we see exactly the contrary during La Niña events. The interesting thing here is that the max N spillover model identified the ENSO cycles, the QQ, as the great, which basically tells us of the places where we see a greatest dry-wet differences in triannual variability, as those were hotspots of Ebogeli transmission. And our time series approach then showed us that probably what we see is ENSO-driven hunting peaks that contribute to outbreaks of Ebogeli spillover. So this is my final slide, the conclusions of the paper. The first one, the most basic one, is that we derived the first detailed maps of polycystic echinococcosis, and we derived them separately for the sylvatic circulation of the disease and for the spillover to human. A very interesting result was that the human model incorporated the animal model results. It was trained with completely different data and so on, but this is something natural because for humans to get infected, we need sylvatic circulation of the disease. So it's natural that some of the information is naturally transmitted to the human model. Our key and most important result is that polycystic echinococcosis is a climate modulated disease. And we show that how and why. First, we show that PE's, polycystic echinococcosis, sylvatic circulation is favored by climate stability, let's say temperature and precipitation, to what we could see. But then we also saw that the spillover risk is affected by these ENSO-driven disruptions of hunting practices. The interesting thing is that results are generalizable here to other zoonotic diseases which are associated with hunting practices in Panamazonia because we identified the concrete mechanism. If you want to put this in a broader context, it's important to recall that ENSO extreme events are intensifying, and they are projected to intensify even more due to global warming. Therefore, we can infer that climate change is indirectly driving the spillover of E. borrelii. How? By amplifying ENSO variability. Okay. And yeah, finally, well, this was quite a methodologically challenging paper because of the nature of our data. Analysis on the temporal domain, at least of infection data, was initially impossible. But given that some of the covariates had temporal information, the climate, the hunting, we could explore some of these patterns, identify some of the potential mechanisms, and complement the static results provided by the MaxN. And well, I leave it here. I'm sorry if I went a bit too fast, but thank you for everything.